Hey, boys and girls. We are here with the acclaimed, prestigious Dimiter Kunnev. The acclaimed Dimiter Kunnev to talk about your talk out at the conference in Bulgaria, right? So, Sofia, Bulgaria?
Yep.
Sofia.
Sofia.
Sofia, okay.
Sofia.
Sofia.
Sofia is the Greek word for wisdom.
Ah.
And it's happened to be Bulgarian's capital. The capital. So that was the actual.
Okay.
The actual
Yeah.
meeting life in the universe.
And there is a, I'm going to put a link out to that YouTube channel, International Physics of Living Systems, where they have all the videos, right? All set up.
Yep. So if you find it, I have also, if you don't have it.
Yeah, I got it. Let's see here.
Okay.
This right here.
Give
International.
it
Let's see here. Why can I not? Oh, let's see here. Yeah, problem. Oh, I know what I want to do. I'm going to do this one. Okay, there it is.
I post this one.
Okay. And what did you think of that whole thing?
So actually first of all was extremely well organized. I mean, I was amazed. So how good was. So it is funded by National Science Foundation. And it was organized from Harvard, Dimiter He was Origin of Life Initiative director there. So, and we gathered together. There was every day there was a speech, talks about afternoon. And afternoon, everybody was free to go and enjoy the life, the Sofia life. The food was absolutely fabulous. The hotel was, I don't know how many stars, but it's very nice in the middle of Sofia. There was also a cultural cultural walk. And you can enjoy many historical places. I mean, Sofia, it's absolutely wonderful, modern capital of Bulgaria. I've been living there probably around maybe 7, 8, maybe 10 years.
Oh,
before
so you went home, kind of.
Yep. So I mean, my hometown is Devin in Rhodope Mountain, so southeast of Sofia. But so I went there, of course, after that. But during the actual presentation, I mean, during the entire symposium, I met pretty much everyone. Unfortunately, John Sutherland, he was sick at that symposium, but I met Dimiter Sasselov. We discussed with him. John Sutherland was sick, but he gave his talk via Zoom.
Yeah.
And it was pretty much identical talk to whatever he presented in Chicago. So
YouTube
his
thing.
talk, however, in Chicago, his talk was kind of more, because probably he get more time, so he presented more detailed presentation.
Did he
And my
mention?
presentation, which, yeah.
Did he mention when he's going to publish a paper on that work that he did or talked about in Chicago? Are they?
Unfortunately, I was not able to ask this question. So
Okay.
he was so excited by my question that he pretty much canceled my second question that I wanted to ask him. So yeah. So I will present at the moment my presentation just to get a view what is important for Origin of Life. But I want to mention several names which are very important to look at. So the first very important name is David Catling.
Okay.
He's from Washington University. He's pretty much a chemist plus geologist. So, and he's the guy that discussed a lot about the prebiotic synthesis, cyanide, the conditions, prebiotic synthesis. It was very interesting, his talk about how reducing atmosphere or not reducing atmosphere was definitely that. Probably next time we just have to go only on his lecture. And immediately after his lecture, Paul Rimmer, Cambridge University, which is from the John Sutherland's lab, also additionally added the stuff to that. So these two must be watched, David Catling and Paul Rimmer. Ben Pierce, he was about cyanide production, but he's kind of more like trying to simulate more things in the cyanide. But anyway, so David Catling, remember this name, he's important, and John Sunderland, of course. There was many geologists, mostly was geologists. Who else? Nick Tosca, he was talking about iron and iron deposits. And he also was discussing about iron cyanide and those kinds of things. It was very interesting, prebiotic. So mostly from the symposium, I will say it's mostly prebiotic symposium. And there was just a little bit about evolution of ribosome, which has been presented from also Matthew Pauner was, but Matthew Pauner, he presented something about possibility of peptide formations. So the evolution of ribosome is Anton Petrov. He is a part of the Loren Williams lab. And also I talk with him, he will come to our live show and he will talk about the evolution of ribosome. So he will be next superstar in this live show because he indeed knows pretty much everything about the ribosome. And I'm very happy that I discussed with him a lot about the stuff. And that's kind of important. Loren Williams, he didn't present much of his work, because actually from his lab, Anton Petrov, he presented the work from his lab mostly. Loren Williams was kind of more philosophically presented. His talk was more like how we should not believe in pretty much all of our hypotheses because they anyway are wrong. And we have to be very cautious about whatever we're saying. But he was kind of very skeptical about the many things that the chemists people said. However, he's very good in ribosome evolution, and of course he's kind of pioneer in the RNA peptide world. So David Catling, Anton Petrov, Dimiter Kunnev, this is the names that you have to watch for sure. And of course, John Sutherland. Paul Rimmer also. Dimiter Susilov from Harvard, he presented mostly like a program from Harvard Origin of Life program. Pretty much that was his that was kind of more bright and interesting talks. Geologist was also very active. They said a lot. And probably we should start actually make some sort of series of live shows to watch just some of the chosen, some of good talks in this particular program and this symposium. It was interesting. So I enjoyed a lot. And I hope someone was listening well. There was a debate after that, private discussions. And I think they got it. Some of them got it, especially David Catling. He was kind of old-fashioned initially about the thinking about the RNA world, but when we discussed together, he realized that actually RNA peptide world is kind of more prone. It's more viable concept. Okay, so
before you go on, I just noticed that there are transcripts available, Dimiter, for some of these talks, like David Catling. He has a transcript, so that'll help a lot, with the audio. So anyway, so we could easily go through those, but not all of them have transcripts. Yeah. But anyway, okay, go ahead. Go.
So let's try to share. Can I share my screen?
You should be able. Oh, you got a transcript too.
Yeah, I can
Good.
share.
You have a transcript. You're roboting. Now you're roboting.
Just one. Am I roboting?
Your audio is real bad right now. I'm not sure why.
Because I'm trying to open my window.
Oh, okay. Well, once you get it open, hopefully it'll work.
Okay, let's open first and we'll see. Yeah, there is forces outside that doesn't want me to go and show my stuff. That's why
Sal Cordova. Yeah, Sal Cordova and
Yeah,
Satan
yeah.
are doing that.
They're doing in purpose. For
Yep.
some reason, I don't see my slide. For some reason, so I'm
I
trying to open it, but okay, let's close war.
You should be able to
Yeah.
share with StreamYard.
Okay, yeah. Let's see. Ha! Got it.
There we go.
Okay, guys, can you see it?
Working on it. There we go.
Yeah. So let's try just for a second to put in a sort of presentation mode. Let's see, is it going to work this way or not?
Okay.
What about now? Is it
It's
well visible?
not in not in presentation mode. It's you're still showing the main screen, so you may have to share that presentation screen when you go into presentation mode.
Okay. So what about now? Can you see it
No,
now?
I can see the whole PowerPoint window, not the
Yeah, that's
present.
pretty much what it is.
That'll
Yeah,
work.
I'm not presenting at the moment. Yeah, I'm not
Okay.
presenting at the moment. So
Okay, I'll make it bigger here. So
I'll try to zoom it somehow. Yeah.
it's a
Okay.
little big right now. But
Okay,
yeah, go
let's
ahead.
do it this way.
Okay.
Let's move it like this. Is it better
Yeah.
now? Yeah.
You're clipping off the top.
So,
Okay, yeah, that's pretty big. Okay, that's good.
yeah. That will do. Better.
Yep. Let's see if you're roboting again.
I need feedback. So,
No.
guys, that's the important stuff here. So it's visible well, right? You can see it.
Yes. Yeah, we can see it
Can
good.
you see my
Cursor too, yes.
Can you see my pointer?
Yes.
All right, good. Okay, let's start it. Okay, guys, so look at that. If you just look all domains of life and try to analyze, there are several papers. One paper is from the Professor Paul Hicks, and another papers, there is several other papers which try to figure it out how many genes are common in all domains of life, eukaryote, archaea, bacteria, and so on. You can see probably between 300 and 400 genes are the common genes for all domains of life. And there is another paper also. This paper is interesting. The paper is from Enrestu Perez Rueda in team. This paper shows that if you look all the proteins, around 52 protein domains, we're talking now for protein domains, are common for all life forms. So based on this, of course, the genetic code is standard genetic code. We can say, and we are sure that there was a last universal common ancestor. So based on that, of course, huge phylogenetic investigation study, just this sentence here, 500 papers. If you go however, to approach from top down, and now you have to look for the paper from Gustavo Honores. He's the guy that presented the very first domains, the very first protein domains which occurred. So he did a very complicated but huge study about all of the domains, and he chose about, I think, 6 different criterias to analyze it. And finally, he figured it out that one type of domain, which is called C37, P-loop domain, is one of the most ancient domain. And also C2 domain, which is not P binding domain. So everybody is familiar with NAT P, NAT phosphate. This is the redoxy system. So those domains which interacts with these dinucleotides are among the most ancient protein domains. And the C37 domain, which is the most, even the ancientier domain, it's interacting with nucleotides. So the very first domains which actually set up and occurred protein domains are those which interacts with the nucleotides. And that's a very interesting. From the study which now Loren Williams, and once we invite Anton Petrov also, he will discuss in details, but the very first structure from the long ribosome is the PTC center, peptidyl transferase center, and also from the small subunits, it is some kind of helices, which is helix 30, it was helix 27, helix 28, and helix 44. So this is kind of joined several helices, which is the most ancient, the recognition between the mRNA and the tRNA, where it's actually started to recognize each other. And the tRNA is very well known to be the most ancient one. So taking all together, you can make a conclusion that the translation was among the very first process of the origin of life, because somehow those ribosomal structure are the first one, and also for some reason, nucleotide binding domains was the first that established, and we have to keep in mind that. That's very important. Any specific question about this? Anyone, something to discuss about that?
Nope.
So I think it's very clear what I tried to say. So, yeah. And if you look from the prebiotic chemistry, so the entire symposium pretty much was about the prebiotic chemistry, based on many, maybe 2,000 papers by now. You can conclude, and lastly, for example, the presentation of John Sutherland, we can conclude that the nucleotides and amino acids usually comes together with one pathway, chemical pathways. And that was something which John Sutherland very well emphasized in his presentation. The nucleotides, if you have nucleotides, you for sure also have amino acids. Oh, I would say vice versa, because usually it's considered that amino acids are much easier to be synthesized prebiotically. So he showed that once you have amino acids, sure you will have nucleotides as well. And there is several papers which also shows that once you have nucleotides, they could polymerize. And in that case, you may have short RNAs. So this is kind of basic stuff which we have to keep in mind when we are talking about origin of life. However, it is very obvious, so this is bottom-up approach. It is very obvious that something between those two is the origin of life. However, that's very important, and everybody just forget about it. The structure, like a protein domain here, and also PTC center, A site of the ribosome, or tRNA, all of that structures are result of selection. They are already selected somehow. You cannot just jump statistically and have such huge structures. Nucleotides and bigger than a few amino acids, and they have a specific sequence, of course, cannot arise spontaneously. So we have to look all of this structure already occurred evolution. So that is the question. How it happened? What actually is important to know about origin of life in order to get this? So this is a picture slide which everybody knows. I mean, from our discussion group. But we have to keep in mind that this is just mechanism for information-dependent selection, for Darwinian evolution. How
You're roboting real bad again.
This is something which was kind of the philosophical questions. What? You want to say something?
Oh, you were roboting for a little while, so every now and then you're going to have to repeat because most of the time you're good, but every now and then it goes out, and I can tell you when.
Okay, shall I?
Yeah, repeat the last
okay.
minute.
So
not too much.
That was the 70s. Actually, that was a huge kind of question about life, how it's possible to keep.
You're doing it
and
again.
I will say how you have Darwinian evolution.
Okay.
So basically, this is the central mechanism, the same mechanism by which life keep positive, negative entropy. So if you have some source, this informational source by some rule determine some structural function, that system either perished or would survive. So you have two different options here. And in that particular case also, the reason for surviving is because this structure of functional component protects the entire system, the informational system. So in that case, this feedback to the protection of the informational source is absolutely vital in that order. So if something is just changing a little bit, because there is a rule determining the functional structure, obviously immediately the informational change will reflect the structural and functional change. And again, in the particular environment, either will be better or worse. Either will survive, will be more adapted or not. that this is the mechanism by which Darwinian evolution actually works. That's why we can say that the life of Darwinian evolution or information dependent selection is a dynamically stable system when an information determinates its supporting structure. So,
Yep.
and that is very important. Now, if you look at that, this particular definition, it's not attached to some sort of particular polymer or particular structure. You can do the same scheme artificially, for example. You can take in the lab and make your own polymers and do, or you can make it in a computer if you want. We can do it in computer, Mike, together with you. So we'll do algorithm which has this, and we'll put artificial environment in the computer, and we'll try to see how this evolves.
Yeah.
So we can do it.
There's still a lot of steps that
So
are
that's
fuzzy.
important. I mean, this is the core.
Yeah.
Yeah. And that's the core of everything we call life. So there is also rule for origin of life. What does it mean? That mean that you cannot explain just information outside the function and structure.
Right.
I mean, they are mutually interacting, dependent each other mutually. So, in fact, information means something as a pattern, sequence of something which have a function. Otherwise, it's not information. So you cannot explain information without having the function at the same time, and vice versa. The actual functional component cannot occur without informational source. Both are absolutely necessary to be together and to occur at the same pathway, the same mutual pathway. Otherwise, you will not be able to make it naturally. So imagine you're talking about information, just occur appearance of information somewhere outside of them.
So, Dimiter,
And
if
you
you
have
turn
function outside. Yeah.
if you turn your camera off, maybe that'll limit some of the bandwidth because you're roboting quite a bit. People are having trouble.
Okay, so let's try to do it. That might help.
Just hit that stop camera or whatever.
So can you see it now?
Yeah. I see your icon. So let's go ahead and try this.
Is it better?
So far, but we'll see.
Okay.
I think I'm going to have to figure out how to bring you in on Zoom. Yeah, it's better yet. Yeah, seems pretty good. Go ahead.
Okay, so let's keep going. So that's a very important moment here, because during origin of life, the information, the structure, and the corresponding rule are locked in their origin. You have to keep in mind that. So everything so far, many years of scientists that was talking about origin of life was just partially right, partially correct, or completely wrong. Because you have to have this point, this particular system ongoing and trigger somehow in order to get origin of life. Otherwise, you will not get it. As simple as that. And those two slides, this one and this one, are absolute slides. Therefore, you cannot avoid those two slides. It doesn't matter from where you're going for origin of life. It doesn't matter what kind of hypothesis you're going to. You must meet this criteria, particular criteria. Otherwise, nothing will happen. And we have to keep in mind, and I told them in the conference, guys, you cannot avoid this. That's it.
Yep.
It's a law. You cannot avoid it. So
yeah, trying to figure out how
that
this
case,
started
I
up.
put together some sort of very reasonable. Yeah, that's exactly the question. You have to figure it out how it started up. So how it's locked in the origin and structure.
Now we're roboting again. Damn it.
Kind of the. Is it still roboting?
Yeah, no,
Oh,
okay,
shit.
now you're back. Yeah, now you're back. And you also have a delay when I say something. It seems like it takes you a while to hear me. I don't know. We're gonna have to figure out how to bring you
I
in on
don't have
Zoom.
problem with my inter.
Yeah, I don't know what it is.
Okay.
Okay. Let's see.
So
Yeah, basically
maybe,
how that structures. Go ahead.
maybe Cordova is watching us. Maybe.
Yeah, he's
Sal.
probably tapping out all our resources. So,
Yeah.
but now you're good. Yeah, now you're good. You can go ahead with this slide.
I will try to talk slowly.
Okay.
I'll be slowly.
Well, try to listen. I'll let you know when you're roboting, but I think you're even getting my words in some sort of delay. So, but and then
Well, at the
if you
moment,
just stop, yeah, if you just stop for 30 seconds, then it seems to clear up.
the moment you're fine. Do you want me to switch off and to go in Zoom directly?
Yeah, but I'm not set up to do that right now. But I should have.
Okay, so let's keep going at the moment. Let's keep going.
Go ahead. Yeah, you're good now. Good now. Go fast.
So in that case, just as a mini summary of everything here, reliable hypothesis for origin of life needs to describe probable, meaning statistically probable information structural relationships. Otherwise, not going anywhere. And also must explain the driving forces of selection with this positive feedback. So imagine you have, I've been reading many hypotheses so far for origin of life. And in fact, most of them are very bad.
Yeah.
They just put in order something more simple, and that goes to more complicated stuff, more complicated structures, but it completely lacks of any selection forces. For example, you have to explain why and what is the reason that your more advanced form survived versus the not so advanced form. And that's absolutely important. Most of the hypotheses so far, I haven't seen these details, which is very important. If you don't have this explanation, you're just jumping and doing nothing. You're just imagining some sort of how might go based on something, but in fact, it's not good hypothesis for origin of life.
Yeah.
So you see how important is all of that.
Yes.
You cannot avoid that. I mean, There is no other way. Origin of life hypothesis must explain genetic code. How the genetic code comes in. Genetic code cannot come later. Usually the very first, for example, RNA world hypothesis, they put the genetic code later once you have kind of RNA entity. But RNA entity by itself makes it is another problem which must be solved. A genetic code comes later. That's a wrong thinking. The genetic code should be at the beginning of origin of life. Also, as we saw from top-bottom approach, the very first protein domain is a nucleotide binding domain. So you must explain why the very first protein domain is a nucleotide binding domain. There must be some sort of following up your hypothesis. Otherwise, you don't have, something is missing otherwise. Another explanation must be why the ribozyme actually come to existence. So the ribozyme, just for anyone to remind, this is a enzyme entirely built up from RNA. And if you think about why the ribozyme should be selected, since now we know that 99.9% of all of the enzymes are proteins. What happened? Why the ribozymes came to be? I mean, you have to have some advantage to get ribozyme at the beginning. Otherwise, you don't have explanation of formation of ribozymes. Whatever is doing ribozymes could be done simply by protein enzymes.
Yeah.
So we need to know why the natural ribozymes came to be. And that's very important also for good hypothesis for origin of life. And of course, where the energy comes from. That's important as well, where the energy comes from. So this is, and of course, you can put more requirements for origin of life. Whatever you want, describing
Cell
the
membrane.
conditions, describing, yes, all many things, but this is the major few of those, which definitely must be explained. So that's why people should kind of, all of the scientists should focus when they're talking for origin of life, should be focused on these things, these conditions. All right. So let's go over, and I will present one very important hypothesis, or let's say concept for origin of life, which has been updated based on the data of John Sutherland's study. So if you look just RNA, so this is RNAs, it hit the environment, let's say some kind of aquatic environment. You hit it, so you have a process of denaturation of the RNAs, which became single-stranded. But once environment, so cool it down, they will start to anneal each other based on Watson-Crick base pairs. And once they find annealing conditions, they will just make base pairs and they will hybridize each other in that case. So, and that's absolutely important, and also it's very well understood. This is, at least this part is absolutely well studied, because we know that since probably 60s. We know all of that process, and all of the PCRs at the moment works based on this particular denaturation and hybridization cycles. And there's many papers which say that in these conditions could be polymerized. Those particular RNA, they could be um, kind of rearranged, and also mostly from those perspective, the GC simply because they possess more hydrogen bonds.
Yeah.
So in the environment, those will circulate fuller. Now, if you add amino acids again to them, amino acids, some of those amino acids will amino acylate to RNA. Now, I want to mention that there is so far in the literature, there is at least 15 to 20 ways, different ways demonstrated experimentally that amino acylation could occur chemically. So, what is amino acylation? When the amino acid with its own carboxyl group interacts with the hydroxyl group of the ribose of the last nucleotide, usually in a tryptophan site nucleotide of the RNA.
Yeah.
And I used to say that this is the most important reaction in the universe.
Yeah.
Not because the others are not important, but this very important between the information and the structure. So it was demonstrated how it's possible and it's happening chemically. Now, also you should pay attention that once two genes amino acylated, because of the process of hybridization, which could be very complicated or could be very simple, two adjacent, close to each other amino acids might go very closely together, and they can form peptide bond. The conditions to make a peptide bond, in order to do that, you need to overcome the process of hydrolysis. So if you overcome the process of hydrolysis, in that case, you form peptide bond. That's why you need wet and dry cycle as well.
Yeah, Nestle doesn't like those.
Process. So again, there is nothing so. Huh? What do you mean?
Nestle doesn't like wet dry cycles. He says.
So it oh Nestle. Well, so he got to listen David Catling's lecture.
He doesn't like to do laundry.
Just let him know to listen. So David Catling's lecture is important for him. Those two slides are clear from any kind of overview. Any regular person that understands what is RNA and what is amino acid, pretty clear should understand these two slides and how the hybridization dependent peptides are formed. So you have to pay attention also that this is hybridization dependent peptides because occurred from the process of annealing of the RNA. This is not peptide that you smash together all of the amino acids and just form peptide. There's a difference here between one and the other. So what is the nature of these particular peptides? Of course, these peptides are not coded because pretty much statistically every RNA could interact with every amino. Hybridization-dependent peptide will depend on the abundance of the actual amino acids. So, and of course, the most amino acids are alanine and glycine. Therefore, most of these hybridization-dependent peptides will contain mostly alanine and glycine. Some of those, of course, will have amino acids like arginine, for example, which will interact with RNA. So that's what is kind of logical way. I mean, there is no other way. Okay. Recently, John Sutherland, he demonstrated a way of amino acylation, which is very interesting way of amino acylation. He published it pretty much this year, a few months ago. It's a fresh information. He presented that upon hybridization process, you have first RNA, shorter RNA, which plays the role of donor of the amino acid. However, this RNA interacts with the phosphate nitrogen interaction. Now, this particular type of interaction is stable in alkali conditions. It's not in acidic conditions. In acidic conditions, this particular interaction, the phosphate on the 5' end and nitro group from the amino acids, it's favorable only in alkali conditions. However, in particular conditions, there is a bridge forming here where the carboxyl group interacts with the acceptor RNA in a way where the
You're roboting.
Carboxyl group with the same chemically type as we know it from the biology. So in biology, we have this type of amino acylation, not this type with alkali conditions. However, in order to occur this particular amino acylation, you gotta go through the alkali conditions, then you have to chill down the temperature, and then you have to go in acidic conditions. So there's another cycle here, which is alkali acidic cycle.
Yep.
But what he demonstrated, and here's the next slide, this is my drawing based on his presentation in a Chicago presentation, and also in the Sofia presentation is the same. He demonstrated that once you have in alkali conditions preparation of the donor RNA, and this donor RNA, it's happened to be specific sequence and transfer in an acceptor RNA in acidic conditions. It's happened that this transfer of amino acids is dependent on the sequence, which is coming somewhere here.
Right.
Let's say a few nucleotides down. This particular phenomenon, which he will publish, it's not published yet. It's a stereochemical mechanism where you have a specific amino acylation based on a sequence of the donor acceptor RNA. And this is very important because in order to form genetic code, you must have some sort of dependence between the sequence of RNA and also the particular amino acid.
Yes.
And Yeah,
That's the most important part, is that the genetic code seems to fall out of this little sequence that Sutherland found, and it resembles
exactly.
the genetic code we have, and that is so significant that I would say that that is like the anti-God particle, because that shows you right there that there's a stereochemical reason for that code to be the way it is, at least for the six or so that he found. Uh, so this is a
Now
really
you
big.
have absolutely, that's a big finding. I mean, that's a huge finding. Also, I want to say
You're
something very important.
roboting.
This is not today. However, you still need the specific stereochemical interaction here. But the
Yep.
genetic code, and I will explain why the genetic code occurs down here on the bottom of
Okay,
this acceptor RNA. This
that's
is
the
some
part I'm
kind
losing
of
sleep over.
proto. Yeah, so I will explain that because if you remember, I don't know who. Yeah, anyway, so there is a two giants in the genetic code formation. One giant is Woozy, and the other is Francis Crick. So Crick said that the genetic code is frozen accident happens.
Yeah,
Woozy said, no, you must have stereochemical reason for
right.
genetic code formation. So this is two huge giants. And they was fighting each other constantly, at least in the minds of scientists. However, with John Sutherland, he and Dimiter Kunnev also, it seems that both are correct. And
Yep.
I will explain why both are correct. So let's combine everything so far in this diagram here. This acids and short RNAs.
Okay, you're
Also,
roboting. So
you
repeat
have
that.
alloy conditions where you have donor RNA. Am I roboting?
Go ahead. Okay. You're back now. So go ahead. Repeat that last little part.
Okay. So we're combining everything together so far in this chart or this RNA, short RNA, and amino acids. In alkali conditions of donor RNA, as I explained here.
Yep.
This particular way. And as I explained here, donor RNA with specific sequence.
Okay.
So this is, and this particular stage in alkali conditions. So clear so far, right?
Yep.
Now, in acid conditions, you have transfer through the acceptor RNA. As a result, you have amino acylated RNA sequence of donor acceptor interactions, stereochemical interactions. If it's not clear, let me know.
Okay.
I think so far so good, right?
So the bottom part would be the frozen
Now,
accident.
don't rush, okay?
Okay.
Step, just stop, just listen. Yeah, of course you're right, but just listen. So now, see these steps here. E, F, G, and H.
Okay.
These steps here. Step is like here. Here. In the where you need hybridization.
You're roboting again. Damn it.
So that's it clear?
No, you were roboting for
Hey
the
guys,
last
I
minute.
need feedback from you.
Yeah, now you're back. You got to repeat the last minute again.
Okay. Okay, so these steps here. So we have amino RNA.
Yep.
Now we have for this four here on cycle, as I explained here, up and down, temperature cycles, right?
Yep.
Okay, so in that case, based on these steps, you're producing peptides, hybridization-dependent peptides. Correct?
Yep.
Eho, just say yes, something say. I need to know that everything I'm talking, someone is listening, right?
I'm listening,
Now,
but
here is the, yeah. What's wrong?
go ahead. There's always a gap in my understanding when you go through this, but if you go through it enough,
Okay, say
I'll okay.
Let's go.
Okay.
Ask it.
If I could coherently ask a question, I would. But okay. So we got EFGH, and they are creating this bridge peptide, right? But let me see
They
here.
create many peptides randomly.
Many peptides. Okay, random. Okay.
They create many peptides randomly.
Okay.
So those peptides are just peptides based on the whatever I described here as the nature of HDP.
Okay.
So once now, of course, they are random peptides to some extent. They are random peptides. However, some sequence, by chance sequence, will interact with particular nucleotide amino acid combination. So it will facilitate, let's say, it will facilitate the process oscillation. And in that case, will bypass the step which is the alkali condition step.
Okay.
Am I clear here?
Yeah.
So you have a specific sequence which bypass and make easier to do. And this bypass is happening because of nucleotide binding ability of this particular peptide.
Yep.
And
that's the bridge.
Why you have facilitation. That is the bridge peptide.
Yep.
And also, in addition to this, which is not shown here, the interaction of the peptide with RNA stabilize this RNA and peptide. They became more stable. This is based on the paper of Williams, Lauren Williams. Okay. So I think it's understood by now. So
I'm getting
if you have
closer.
this bypass, getting closer. If you have this bypass process, in fact, this is a driving force of facilitating amino acylation and bypassing this particular aldose to run.
Yep.
Understand? And that's a huge selection force. Also, in the future, this particular peptide will become longer and will be fit better and better every time. So it will start to adapt better and better because we will facilitate and better the process of amino acylation. And in the future, this peptide developed into amino acyl synthetase.
Yep.
After many, many, many steps. At the same time, the process of interactions will start to co-evolve. The small and the big subunits of the ribosome will start to co-evolve each other.
Yeah.
And to become better and better. Now, what is the important point here is, again, independence from the wet and dry cycles. So from one side, you have independence from the process of the RNA. From another side, you have independence from the cycles, wet and dry cycles, by evolution of the ribosome. So this, we call it Terra-Darwinia. Terra-Darwinia by default is the first chemical system capable of evolution. So capable.
Okay, now.
Of Darwinian evolution. Darwinian evolution is what it makes the information. So the information comes because you have stereochemical interaction between the amino acid and the acceptor RNA. There is a rule which determines formation of peptides. The rule is Watson-Crick base pairs plus a very primitive genetic code. Now, I'll go back here just for a second, and what you said, Mike, here is that the frozen accident is because just for a few amino acids is the frozen accidents, because only some combination of this bridge peptide will work.
Right.
Therefore, you have a frozen accident, just a few, 3 or 4 amino acids got to be frozen accidentally in this particular code.
So that the bridge peptide can be formed over and over again.
So you have stereo. Can you repeat back?
So that the bridge peptide can be formed over and over again. That's an important part of the cycles. Yeah. Okay.
Exactly. That's the trick. And now if you go and look at that, the positive feedback is actually because of the bridge peptide, as we described.
Yeah.
So you have a process of Darwinian evolution, you have a selection forces, and everything is feasible. It's possible in terms of statistic. This is not huge structures. This is small structures.
Yep.
And once you have this particular system, you have evolution, and everything else could be explained later.
Yeah, that's the
So just a
generation.
summary of so this is just the summary that this hypothesis described the probable information in structure relationships, also explained the driving forces as I showed, explains the formation of genetic code as we saw, frozen accident plus stereochemistry, also explain why the first protein domains are nucleotide binding domains. Understand?
Yep.
And now explains also ribozyme formation. How explains ribozyme formation? It's explained here. So initially, so this is the time frame, the time stage, you have the RNA pterodarwinian evolution, which kicks out and continue. The coded HZP, which is the bridge peptide, now became coded, start to make a small peptide motifs. But you have to pay attention that by making these small peptide motifs, you don't have much functions, because any peptide interacts only with whatever amino acids is there. It's building up this peptide. So you don't have much choice of functions. Therefore, at the same time, ribozymes will be selected as well. That's why the ribozymes are selected so early.
Yeah.
Because you have RNA peptide world. At the moment once the proteins start to develop, meaning you have very long chain of peptide which start to fold and makes 3D structures with the active centers, then your ability of functions became much bigger, high number of functions could be selected for.
Yeah.
At that moment, ribozymes are no longer favorable for selection, but proteins are favorable for selections.
Yep.
And the genetic code is formed meanwhile. So let's talk about the genetic code, but this is pretty much what it is. So I will stop at the moment.
Okay. Should we let those hooligans in to ask you a question? Dessel, Drace, and Immutable, and Nestlig?
If you have questions, whoever have a question, let's discuss, but not very long.
Yeah,
so
I don't want to go
let's
too
have
longer.
A few questions actually.
Yeah. Why don't I let those guys in? See if Immutable, Nestlig, and Dessel want to come in, and we'll spend a few minutes, about another 20 minutes, and I want to wrap it up, okay?
The link is in the side chat.
Okay.
Oh, it is. It's in there twice. I put it in. Anyway.
Hello.
Hey, guys.
Hey.
I put a question a while back, and it was about like I've seen what is called the Darwin eigen cycle, and are you familiar with that, Dimiter?
I'd like the paper on that in the description.
Sure.
Darwin eigen cycle. Can you try to?
Kunnev and Wolf came up, right? Nestlig, was that Kunnev and Wolf?
Yes, I was looking up the paper. I have it. There we go. Actually, they have several papers, but this is one of the papers that they have on that.
So can you try to formulate your question?
Oh yes, the eigen cycle is the eigen threshold specifically is the threshold between the size of the genome and translation fidelity. Like if you have a low rate of fidelity with translation, that limits the size of the genome that you can have. Or
Oh,
at
you
least,
talk about
Yeah.
Eden cycle. No, it's not it's Eden cycle.
or Egan,
I know
Egan's,
about
maybe I
that.
pronounced that.
Yeah,
Yes.
I simply could not recognize what you're trying to. Yeah, of course, it perfectly fit. If you just listen what I said, you will see very well that it's such a small functions, which is just a small system, very easy system. You everything that you need is just to trigger some sort of feedback selection. In that case, it's perfectly fit with this particular concept, and we know about that concept very well. It was long concept. It was around for maybe 10 to 20 years around, because you cannot have but this is based on a very old concept of what actually life is and what the actually life originated is. So even so, there is no at all any controversial or something that it's provocative here. It fits perfectly well. Any other questions?
Immutable had one question. What exactly is it? Is it acts as a rate determining factor for how quickly amino acids will polymerize? Is it based on how stable two RNA oligomers are when they interact with each other? Oh, right.
Two RNA
What exactly?
I'm not sure. Quickly, amino acids will polymerize. My late Sunday brain.
So Immutable is starting to ask a very detailed question, which only experiments can do. We don't know exactly the conditions of primordial Earth. However, whatever I said and whatever I described as a concept, it's based on data from John Sutherland. So you can ask him if you want. However, he didn't show, he showed just this stereochemical aminoacylation is a specific process, and that was very important. He didn't go further in his description, but a rate determining factor, it will be for sure. So one rate determining factor will be the actual cycles in primordial Earth. Could be hours, could be days. It depends.
I believe there was a recent.
Yeah,
I believe there was a. Oh, sorry, do you want to finish the answer first? Sorry.
Yeah, I mean, basically, it's an experimental question.
Like I remember also a recent paper that goes into the there is a core within the large subunit of the ribosome, which is the peptidyl transferase center, and researchers believe that that's the oldest part of the ribosome, and they made a proto-ribosome from that, and it can actually catalyze peptide bonds.
So
yes.
Yeah, this is you have to ask these questions later on when we invite Anton Petrov here in our live show. He will discuss about the actual evolution of the ribosome. However, the PTC center is the oldest from the larger subunits.
Yes.
The smaller subunits have its own oldest structure. Mm-hmm. So, and you cannot compare both, and you cannot say which one is older. Basically, right. They both co-evolved.
Yeah.
Well, I think the evidence indicates that they were once separate structures that later became integrated together.
That's like to me, not exactly. So it depends on the model. So it's all about the model. Evidence, I asked him explicitly for that, but there are lots of things there, so it's not be explained very easy.
Well, it's for me, like maybe I'm wrong, but I've seen that the parts that are involved in the interaction between the large and small subunits, those parts that are required for the interaction are younger than the oldest parts. That's why I think they are they used to be separate from each other. At least I suspect so.
Okay, you got it.
I mean, but also there's also the question
So,
about what were the
I've
points.
heard lots of this, especially from the. I'll go to the DESO. Yeah, I'll go to the DESO question. So, but let's say that lots of hypothesis comes from the assumption for RNA world. And those concepts, they pretty much assume that PTC center was playing different role than whatever it's doing now. But that's a different story. So it's a long story, and we don't have time right now to discuss in detail.
Mm-hmm.
So that's all for the David Catling. I discussed with him, and during his presentation, actually, because of you, and I'm respect your opinion in that case, I asked him explicitly, David Catling, about, and you can see it on the talk. Actually, it is recorded. I asked him about Michael Russell's hypothesis and how is it possible somehow to, let's say, join together these two hypotheses. Whatever David Catling told me, there is no any papers demonstrating, experimental papers demonstrating the things which Michael Russell shows. That was his answer. So about wet and dry cycles, he mentioned several names. However, I don't remember it exactly which one. But that was interesting talk about possibility. And pretty much his answer was based on experimental papers directly on deep sea vents. That was his answer. So we can discuss another time with you exactly about that. So any other questions around? You're muted. Speed.
Speech, you're muted.
Oh, my question was, why are we doing this late on a Sunday evening? Okay, you want to
That's
wrap
a
it?
good question.
Yeah. That was,
Immutable as a question, a new one.
The bottom one here.
Yeah,
I would say there's a reason you can have some factor stabilizing two oligomers, maybe something to do with base pairing or some weird interaction with the nucleotide. Yeah, yep. There's that. But basically, RNAs can be stabilized by peptides, small peptides, and they can. I
and that
think
does not exclude. Yeah, does not exclude lots of metal ions around.
Yeah.
Especially magnesium.
Yep.
And iron as well.
Yeah. God, we have so much to know yet.
Also, I had one remark earlier in the talk where I mentioned some work from Williams, and he shows that like the ribosome is constructed in layers from like the old center and layered from older to younger again and again. And when you look at the proteins themselves within each layer, you can see basically the evolution from non-folded proteins to folded proteins, which was really interesting as well.
I think that was the work of Loren
That was
Williams,
maybe
the latest work.
And Williams, yeah, or Williams, yeah.
Okay, guys.
Yep.
Want to end it? I want to give James one thing about the Magic Daddy. I think John Sutherland basically disproved God entirely. He just shot God in the head. So I don't know. That's my feeling on it. Anyway, guys, should we wrap up? Any final words?
Yes.
Okay, what
Final
about the
words?
cruel world?
Cruel world, yeah. That's all we need.
Goodbye, cruel world.
Yeah, okay, we're gonna end her. Okay, guys, goodbye, cruel world. We're done.
Okay, I will see you after that.